So welcome everyone. Good morning, Arizona, and good evening, Saudi Arabia. Today I would like to shortly introduce the group which is working on stress granules, and hopefully you will find something interesting that potentially we could interact later on. So I would like to start with a short introduction of the group. So we are a small group, we are just starting, we are quite international. So for the moment, there are 3 postdocs who joined my group, that is Nicholas, Israel, and Itzel. And also Daniela, she is a lab assistant. So KAUST, I think I don't need to mention, we are located, as you can see here. And hopefully in the fall, I will get also 2 PhDs, one from China and one from Colombia. So the major interest of our group is to study complexes of small molecules, proteins, and also RNA. And we have 2 directions of our research. The first one is the stress granules. In this aspect, we would like to characterize the foci which are formed under stress conditions and identify stress-specific factors. We would like to also understand the machinery, how the stress granules are formed in plants, and also we would like to focus on few candidates that we discovered to be a part of stress granules, but they were not characterized yet to play this function in plants. The second direction of the group is to identify small molecule protein complexes which are stress specific, especially in plants that are already known to be resistant to different stresses. For example, date palm, which is resistant to heat stress. So now I would like to start with introducing how the story with stress granule started. So during my postdoc, I was mostly involved in developing different tools to study small molecule protein interactions. And we wanted to focus on identifying complexes between metabolites and proteins. So as you know, they are primary metabolites, which are considered to be essential for the living of the organism. And they are also secondary metabolites, which are more specialized. So those can be involved in different signaling. So we were mostly interested to study interaction between secondary metabolites and proteins. For that, we use a method based on the separation according to the mass. And what we observed that when we apply the lysate produced from different tissue of plant or yeast, we could separate protein complexes, but also some metabolites which were present in the high molecular weight fraction. So here I want to show you how such a heat map or the elution profile look like for the metabolites. So you can see that in the high molecular weight fraction, small molecules, so different metabolites were eluting as well from the column. This means that they were eluting there because they have a protein partner. And one of the metabolites that I was focused at that time was 2'3' cyclic AMP. And during the separation of different complexes, we discovered that that molecule, so 2'3' cyclic AMP, is co-eluting with proteins, and one of the proteins which was repetitively coming as a complex with that molecule was RBP47, so the protein which is well known as a stress granule marker. Here I don't want to go into the details, but eventually we describe a model when a small molecule such 2'3' cyclic AMP could induce stress granule formation in plants. So what do we know about stress granules? So stress granules, first of all, they are produced under stress condition. So when, for example, there is a high temperature stress, there is inhibition of translation, and at that point, there is a different selection of mRNA. Some of them are going to stress granules to be protected from the degradation, and some pool of mRNA is going to the processing bodies, which when it's being processed, and there will be decision-making if they should be degraded or also protected. So in general, stress granules, they are non-membranous cytosolic organelles, that are formed, as I said, under different stress conditions. The major role which was described so far is the protection of mRNA. However, recently it was also claimed that they might be involved in recruiting not only proteins, but also metabolites. And additional function which could be interesting for the stress granules is that they might recruit important regulatory proteins. So they might modulate the response at the molecular level. And RBP47 is one of the key components of stress granules. So one of the major questions which was actually bothering, let's say, scientists from very long time is to know the composition of stress granules. And in 2016-17, there was a major work which came out, and actually, as David mentioned, Roy Parker was a leader of this research. So they identify composition of stress granules in yeast and in human cells. And the two major discoveries were that the proteome composition of stress granules is quite diverse, and it contains not only RNA machinery, but also ATPase binding proteins, which might facilitate stress granule formation. In terms of the transcriptome, which they identify, they couldn't conclude any specific subset of mRNA to be enriched. So the major outcome was that there is there might be many different mRNAs located into stress granules without specific, let's say, function. So after seeing that research done, I also wanted to try to translate the protocols and to adapt it to the plant research. And what we did in our lab was to extract stress granules from plant tissue. So for that, we use the line when we overexpress RBP47 under as a fusion with GFP. And we treat seedlings with stress condition to trigger formation of stress granules. After that, we perform grinding and different centrifugation steps. And as you can see here on the left side, the stress granules were formed in tissue, and after centrifugation, we could obtain freely swimming stress granules in the lysate. That was the starting point for the affinity purification when we use antibody against GFP. So after such a purification, we ran proteomic to see what we could catch in the stress granules. And actually the result was quite surprising. We observed many diverse proteins that are involved in many different processes. So we were expecting mostly proteins from the RNA machinery, but in fact, there were many proteins which are also involved in regulation of metabolism, hormonal signaling, stress response, and many other processes. So when we obtained the list of 128 proteins, the first thing we did without having a lot of knowledge in plant field, we compared the list to the list of proteins which were identified as stress components in yeast and human cells. And we observed that 25% of proteins that we identified, they are actually conserved among species. And those are the proteins which are the key components of stress granules. So RNA binding proteins, all the elongation factors, translation initiation factors, also ATPases, chaperones, and heat shock factors. What was the most interesting for us as a plant scientist, we identified many proteins that were never shown to be present in the stress granules. And those are proteins involved in different signaling cascades, hormonal regulation, and metabolism regulation. So based from this discovery, we can say that stress granules in plants, they have really diverse proteome. So the question was actually why those proteins are going into stress granules. What is the function? And there are few potential functions that are being discussed. The first one, which is quite conserved and well accepted, is that stress granules are formed to regulate mRNA storage under stress condition. Second hypothesis, or the second potential function, is the recruitment of proteins in order to transiently inhibit their function or also protect them from the degradation. And the third potential function is formation of metabolites under stress condition. So from our discovery, I would like to show you an example of a protein that might be transiently inhibited by the relocalization to stress granules. So that protein is a cyclin-dependent kinase, which is very important for plant development because it regulates leaf growth. And it was shown that the activity of that protein is arrested under heat stress, but the mechanism, how is it happening, was not known so far. That's why we found it quite interesting. So the first thing which we did was to get the line when there was overexpression of CDK with the fusion protein, in this case was Venus, and we observed if that protein in fact will be going to stress granules under stress condition. And here you can see here that under control condition, the major localization is in the cytosol and in the nucleus, and under heat stress, you can observe a lot of dots which are appearing, and those are stress granules. As a control, to prove that in fact these are the stress granules, we usually use cyclohexamide. So that chemical inhibits translation, which means that mRNA will be stuck on the polysomes and cannot be relocalized to stress granules. So when we have a treatment of cyclohexamide and then heat stress, if there is no stress granule formation, it means that the granules which are supposed to form are based on mRNA. So the next step which we decide to check was, of course, to co-localize it with the stress granule marker. In this case, we did a transient transformation with RBP47 fused with RFP, and you can see clear co-localization of these two proteins. One of the features of stress granules is that they appear under stress condition, and they have also dynamic behavior when stress disappears. So what we did, we decided to check what is happening with those stress granules when we take off the stress. So here at the time 0 is sample when we have stress conditions, so heat stress, and then we move plant to normal room temperature condition. And we follow the localization of CDK 10, 20, 30 minutes up to 120 minutes to see what is happening with the foci. And you can see that with the time already at around 1 hour, 1 hour and a half, stress granules start to disappear, which suggests that the formation of those granules is quite transient. So based on that, based on our experiments, but also on the knowledge which is already available, we came out with the suggestion of novel mechanism for stress granule dependent regulation of CDK activity. So under non-stress conditions, CDK normally promotes growth of the plant. When there is heat stress, so high temperature, CDK is being inhibited by moving to stress granules. And then plants decide to not grow and save energy. When the stress is gone and plant enter to the recovery phase, CDK is released from the stress granules, which eventually also disappear, and plant can restore growth. So that is an example how proteins might regulate their activity by being triggered to stress granules. So here, as I mentioned before, we identify 128 proteins, which were cytosolic proteins. But what is important for us that we also obtain 28 plastidyl proteins in our pull down. At the beginning, we thought it's not interesting or maybe it's artifact, but we took a closer look. And in fact, there were many proteins that had potential to interact with RNA, and they might show the properties as proteins which might form stress granules. So we decided to have a deeper look on that. And one of the proteins which we decided to focus on was a snowy cotyledon. That is a chloroplast elongation factor, so it's involved in regulation of translation in the chloroplast. And that protein is very important because any disturbance in the function of that protein might cause delay of plant development, and actually plants are suffering a lot, so they cannot grow as usual wild-type plants. So we decided to use that protein and perform the same stress granule extraction, but focusing this time on plastidial stress granules. And here, once we obtain a stable line expressing snowy cotyledon with GFP, you can see that under heat stress condition, it formed the stress granule, but inside the plastids. Next thing to focus was to check how is the dynamic of the formation of those plastidial stress granules. And here again, we follow the dynamic after the recovery or under the recovery phase. So after applying heat stress, we move plants to the normal condition, and you can see that with time, the granule also disappear, showing again that they are very dynamic. And another test which we perform to characterize more the properties of observed plastidial stress granules, we decide to check how stable they are. So there is a chemical, hexanodiol, which is known to dilute or to dissolve the transient interaction from the shell of stress granules. When we apply that chemical and we follow the stress granule in the time scale, we observe that the area of the stress granule is getting smaller, but they don't disappear. That suggests that, in fact, the plastidial stress granule that we observe, they have a shell which might be dissolved, but they also have a stable core that is being there and is more, the interaction between its components is more stable. What we did next, we focus on characterization at which temperature and at which time the plastidial stress granules can be formed. And here you can clearly see that half an hour of around 33 degrees is enough to observe plastidial stress granules. So after this short characterization, we decide to extract these plastidial stress granules. For that, we use again the protocol which we prepare for the cytosolic stress granules. And here we identify around 88 proteins and also 4 metabolites. And again, there was a very high diversity of the proteins. So there were many proteins from RNA binding, translation. There were also proteins involved in chlorophyll synthesis, photosynthesis, metabolism. What we did for this experiment, we also performed RNA-seq. And here we observe a high enrichment of mRNA that are encoding genes involved in photosynthesis. And that was very interesting for us because, as you might know, under heat stress, photosynthetic machinery is one of the most sensitive parts. So many proteins are being degraded. So we think that one of the mechanisms that might be happening under stress condition is that mRNA from exactly those proteins will be protected in the plastidial stress granules to be fastly released when stress is gone and rebuild the photosynthetic machinery. So after that, of course, we choose a few candidates to also confirm that the proteins that we are finding as a potential stress granule components, that they are in fact localizing into stress granules. And here you can see that we could confirm 4 of them to be present in the plastidial stress granules. So for that project, the next steps would be to follow the dynamic of the plastidial stress granules for these different candidates that we obtain, and also focus on highlight as that stress is the most relevant for the machinery in the plastids. So now I want to just shortly mention the future direction of the group. So we would like to focus on identifying different composition of stress granule from different environmental stresses. So we believe that depending from the stress, there might be different stress granule form, because different proteins, different transcripts, and different metabolites might be recruited, which is also important for triggering the proper tolerance of a plant. The second aspect would be also to prepare a map of stress-related protein metabolite interactions. And here we would like to focus on stresses which you can see here. So low oxygen, salt stress, heat stress, and drought. And as I mentioned before, also important is to focus on plants which have already known resistance, for example, to heat stress or to low oxygen. So one of the projects which will be running or which are already started in the group rely on the identification in more details of the proteins which are localizing in the stress granules. So there is a big part of proteins that contain prion-like domain. That is a region which gives a special property for a protein to aggregate and form droplets inside the cell. So there was a paper from last year which showed that actually this prion-like domain might define the properties of a protein to become a thermosensor. So here you can see the ELF3, so early flowering tree protein in Arabidopsis, contain prion-like domain with polyQ repeats. What authors did, they also compare if other species, plant species, contain the homologue of that protein, and if the prion-like domain is the same or there are any changes. And as you can see here, when you have the comparison of the sequence, grass, so Brachypodium, they do not have prion-like domain in the sequence of the ELF3, and tomato has a very small potato has very small prion-like domain. So what authors realized that when plant possess this prion-like domain in the ELF3, it responds to the change in the temperature. So usually Arabidopsis needs a warmer temperature to induce flowering. So when the protein has a prion-like domain, it responds to the temperature and can start flowering. However, when Arabidopsis did not, so when the Arabidopsis was transformed with the ELF3 without the prion-like domain, it was not able to respond to the temperature. And that is because the version of ELF without prion-like domain is not able to form stress granules, so it cannot form the aggregates. So that is a very important function for a protein to give the response to temperature. So we would like to follow that direction, and we have already few proteins that actually show to have prion-like domain, and they were not identified as important in the response to stress. So we have a list of 14 different proteins, and now we would like to focus on that and characterize them by playing with this domain and check how much it affects the response to stress and the tolerance to stress. Another aspect which we want to study is to understand the machinery of stress granule formation. So in human cells, it was recently shown that methylation of mRNA is very important for assembly of stress granules, and we identify two proteins which are being located to stress granules, and they are readers of methylation. And also different groups at the same time report that they might be localizing to stress granules. So those proteins are very important because they might affect the development of the plant. So any disturbance in the function of those proteins is really delaying the growth of plants. So we would like to understand if methylation is important for stress granule formation and how much it can affect the stress tolerance. So the last direction which I just want to mention is that so far it was only believed that stress granules are formed when there is inhibition of translation happening. However, just last December, scientists proved that in human cells, some of the transcript might actually be translated. So for that, they use a fluorescent hybridization in situ when they exactly fish the mRNA of a transcription factor ATF to be localized in the stress granules. And then using SANTAC technology, they could prove in vivo even that single molecule of mRNA is being translated. So now our group is working on translating that method to the plant field and prove if stress granules also have transcripts that are being translated under stress conditions. Okay, with that, I would like to finish my talk. There is many people I would like to thank. So first of all people from CDA, I feel really happy here and I'm glad that I could join you. Many collaborators from around the world, many students and people who helped me to work on my research. I'm really grateful to my group, which maybe is small but really motivated, and of course to my family that keeps me busy but also motivated every day. Thank you very much for your attention.
Thank you, Monika. That was a fascinating talk. I really liked it. As I mentioned before, we're going to hold questions until after the second talk, and I have to confess that I did forget to mention fun facts. Both the speakers were asked to provide fun facts about extra things. And so in Monika's case, she's very enthusiastic about sports, the gym and in running. And she's also addicted to travel, as are many of us. And she loves animals. And so, Monika, thank you once again. It was a fascinating talk. I really enjoyed listening to you. Okay, our second speaker today is Jesse Woodson from the School of Plant Sciences. The topic of his talk will be control of photooxidative stress by chloroplast quality control pathways. He's an associate professor in our school here. He received his PhD in microbiology at University of Wisconsin, looking at vitamin synthesis in the archaea. He was a postdoc at the Salk Institute within Dr. Joanne Chory's lab. He worked with her towards understanding the complex relationship between chloroplast organelles, photosynthetic activity, and cellular signaling in plants. Came to us in 2018, and his group investigates how chloroplasts can sense their environment and signal to the cell for acclimation. And I'll remember to announce his extra things. He tells us as a high school student, he was a semi-professional juggler, but he traded in the glory of that career for a career in molecular biology. So Jesse, I'm looking forward very much to your talk, and I can see almost immediately the synergy that you could have with Monika and her interests. So thanks again for agreeing to talk, and I look forward to hearing your story.
Right. Yeah, it was a difficult decision to decide between the career in the circus or in the science. But I made my choice and I haven't looked back. Well, thank you very much, David, and thank you all for coming to listen to our talks today. So yes, as David said, I'm gonna be talking about what my lab has been doing since 2018, which is trying to understand how chloroplasts can sense their surroundings, sense a plant's surroundings, and turn those into signals to regulate or change plant physiology. And one thing we're understanding or realizing is that there's a lot of signaling chloroplasts do with the cell, and some of these signals actually direct cellular degradation and can lead to chloroplast degradation and chloroplast turnover. And I think that's nicely illustrated in this EM micrograph I'm showing you here. This is an Arabidopsis mesophyll cell with 4 chloroplasts, false colored in green. And this one right here on the top left has suffered so much photooxidative damage that the cell is targeting it for turnover in the central vacuole over here. And so we're trying to understand how this works. What are the mechanisms involved? How is this regulated? Because not very much is known. Okay. So I'll just begin very briefly, just talking a little bit about abiotic stress in general. So obviously stress is important in trying to think about agriculture. Over here, I'm showing you maximum yields of important crops in the United States and around the world. And so you can see what the maximum yields could be and how much we often lose due to various types of stresses, abiotic stresses in orange, biotic stresses in yellow. And in this particular graph, they've lumped everything together. So when they talk about biotic or abiotic stresses, they mean everything like drought, changes in temperature, like heat, excess light, salinity. So you get the idea that these in general can lead to big reductions in yield. But the thing I want to point out is most of these stresses, if not all of them, lead to photooxidative stress in the chloroplast, which is part of the reason that plants respond the way they do. And so we hope that if we can understand why this leads to photooxidative stress, why plants respond this way, maybe we can find ways to mitigate this level of yield loss. Now, by this graph, you might think, well, plants are pretty lousy at dealing with stress. It's actually the opposite. They're really good at it. They just don't necessarily respond the way we want them to. They often respond by not growing. But plants can't move. They're sessile organisms. So, of course, they're really, really good at dealing with stresses at a molecular genetic level. And what we're trying to understand how they do this. And I mean, arguably, we've been trying to understand this for 10,000 years, how plants respond to stress. But more recently, with molecular approaches, we're understanding that they have lots of different mechanisms. And one of the major ones is that they use their energy-producing organelles to sense changes in the environment and then signal for acclimation. This is how a lot of eukaryotic organisms work, using the mitochondria. Plants use their mitochondria, and particularly the chloroplasts. So the way we think this works, not just in plants, but in just eukaryotes in general, is that there's some sort of stress or stimulus that leads to a change in metabolism in the chloroplast or localized damage. And then here's the mystery part, that's turned into a signal that then signals to the cell for acclimation, and what we call usually retrograde signals, and the best studied controlled nuclear gene expression. And these can lead to really big changes in cell physiology, choices between growth and defense, changes in metabolism, photosynthesis, cellular degradation, cell death. That's what I'll be talking about today. And in the case of animal mitochondria, these signals can affect aging and tumor progression. So the reason I think organelles do this is because they make a lot of reactive oxygen species, particularly chloroplasts. So that includes singlet oxygen at photosystem 2, superoxide and hydrogen peroxide at photosystem 1. And that's on a good day. You start adding stress, they make a lot more ROS. And ROS is bad. You get oxidative damage, you oxidize lipids, proteins, DNA. That can lead to photoinhibition, growth inhibition, or cell death. And so at the very basic level, these signals need to be in place just so the cell knows what kind of ROS damage is going on. But at the same time, this is information. So if a chloroplast is cruising along and all of a sudden it starts making more ROS, that means something has changed on the outside, something in the environment or the situation for the plant has changed. And so it shouldn't be surprising, perhaps, that a lot of these signals regulate genes that have nothing to do with photosynthesis or chloroplast biogenesis, but things like flowering, leaf development. Now, these retrograde signals have been studied for a long time. First paper I know of is in 1979 that posited that there's a single plastid factor that moves back and forth or moves from the chloroplast to the nucleus to regulate gene expression. Probably a lot more. We know it's a lot more complicated than that. One review cited 40 different signaling molecules being proposed. And we still really don't understand any particular one that well, but generally we think that they signal collectively. So all the chloroplasts within a cell sort of get stressed together, send the signal to the nucleus to make this broad change in nuclear gene expression with the point of making photosynthesis more efficient, reducing stress, reducing ROS, or mounting a defense response. And these are still very important. There's still a lot to understand about these. But when I was a postdoc with Joanne at Salk, we were thinking about, well, these are useful signals, but what happens if only a handful of chloroplasts get damaged in a cell? So a mesophyll cell can have over 100 chloroplasts in it. So let's say only 1 or 10 get damaged. What is it going to do? How is a nucleus going to be able to differentiate between all the different chloroplasts? And at the same time, this isn't something you could ignore because this chloroplast could make lots of ROS that would then potentially kill the cell, lead to cell death. So it made sense to us that there's some sort of chloroplast quality control mechanism to protect or remove damaged chloroplasts from the cell. Now, there's actually been evidence for this for a long time from microscopists. So in the early 70s, it was shown that chloroplasts in pumpkins of all plants get degraded selectively during senescence. And also aberrant chloroplasts that don't develop properly were shown in Arabidopsis to be shuttled to the central vacuole to be turned over. So we knew this happened, that chloroplasts get turned over. But it's really only recently that sort of this field of chloroplast degradation has emerged to try to understand how this works, what are the mechanisms, and what regulates it. And it turns out it's actually quite complex, which I guess shouldn't be surprising. This is not something that should be taken lightly by a cell to start degrading its organelles. And so it turns out there's lots of different pathways. Some are autophagy dependent, some are clearly autophagy independent. And some are induced by stress, some are induced by senescence. So we think that the point of these pathways is to maintain healthy population of chloroplasts, but also to recycle nutrients. So chloroplasts contain about 80% of a leaf's nitrogen content. And so if you're thinking about moving nutrients from sink to source tissue, chloroplasts would be a main target for degradation. But we still really don't understand how these work. And so the big questions in the field are, well, how does it work? What are molecular mechanisms? How do individual organelles communicate with a cell? How are they targeted? How is any of this regulated? And what we hope is that if we can understand it, then we can control it and perhaps have some tools to develop stress-tolerant plants. So in my lab, the way we study this is we use a Arabidopsis mutant called ferrochelatase 2. This is a mutation affecting this enzyme in the chloroplast right here that makes heme, FC2. And what happens in this mutant is at dawn under diurnal light cycling conditions, this pathway, the chlorophyll biosynthetic pathway, is activated and the substrate of FC2 accumulates very quickly. So that's called protoporphyrin 9. You see in 10 minutes, 45 minutes, it spikes. The problem with proto-9 is that it's a very photosensitive molecule and it instantly makes singlet oxygen. This is actually quite similar to the very rare disease protoporphyria that some people have that leads to a skin sensitivity to sunlight. It's because you have a problem in your mitochondrial pathway for making heme and you accumulate protoporphyrin 9 and you make singlet oxygen in your cells when you're in the direct sunlight. And we can measure that. So here's singlet oxygen stained with singlet oxygen sensor green, which fluoresces. We can see that you make a lot of singlet oxygen in the FC2 mutants when you're growing it under these light cycling conditions. So what happens is, well, the chloroplasts degrade. And so if you grow your FC2 mutant under cycling conditions, they bleach out and die. But this is conditional. You can grow them in constant light where you don't get that huge amount of singlet oxygen, and they are relatively healthy. And you can watch these chloroplasts degrade. So this is a FC2 mutant that's expressing a rubisco protein with YFP on it. So rubisco is normally in the chloroplast, and you can see that YFP tag or label chloroplast right here. You turn on the lights, the chloroplasts start to swell up, they degrade, they spill out their contents into the cytoplasm and vacuole, and by 8 hours they're gone. So here we have this mutant. You just turn the lights off, turn it back on, and you get this burst of singlet oxygen. You can watch chloroplast degradation, but all the cells die. So we also have looked under these permissive conditions that still make a little bit of protoporphyrin 9, a little bit of singlet oxygen. And we see there actually is still chloroplast degradation there. So here's a cross-section of an FC2 cotyledon grown in constant light. And we're going to zoom in here. And you can see everything looks pretty normal from a distance. Cells are healthy. You see lots of chloroplasts. They look relatively normal. But you look a little bit closer and you start seeing degrading ones. There's a degrading chloroplast here. Here's a degrading chloroplast here. Here's a degrading one that's actually being pushed into the central vacuole, presumably for turnover. Here's some high-res versions of that. So most of the chloroplasts in FC2 look pretty normal, pretty healthy. And then some of them, about 20% or so, it really depends on what you're looking at, but the minority, but still quite a few, are degrading or in some sort of level of degradation. And a lot of them still have starch granules. So we do think these were doing photosynthesis at one point, and now they're being degraded by the cell. We can quantify this. We certainly see more degrading chloroplasts in the FC2 mutants. And then every once in a while, kind of rarely, we would see one being pushed into the central vacuole. And what's, I think, striking about this image is that everything else in the image looks pretty healthy. So the chloroplast next to the degrading one is healthy. There's mitochondria right next to it. They look healthy. The cell next to it looks healthy. So to me, it looks like this chloroplast is being specifically targeted for degradation by the cell. So we wanted to get a better image of these structures. So we took a three-dimensional approach, three-dimensional EM approach. This was a collaboration with James Fitzpatrick at Washington University. This is work done in part by Karen Fisher, who's in my lab. And so we did three-dimensional imaging with EM. So this is what the 2D image looks like right here. You can see here's the degrading chloroplast blebbing. That's the term we sometimes use, blebbing into the central vacuole. This is what it looks like when we reconstruct it. And what we learned is, well, what we verified what we thought or assumed was that this structure in red, this bleb, is completely in the central vacuole. You can actually sometimes see protein being moved into it. The size is quite variable. It can actually, we found instances where it's twice the size of a chloroplast, although this, what I'm showing you right here, is about the average size we usually saw. And they're much more common than we thought. So over here, there's a quantification of how often we saw them in the FC2 cells. About 8% of chloroplasts were doing this, or a third of those that were degrading. And so we think the reason we saw so much more in the three-dimensional is because the contact point between the chloroplast and the bleb is very small. It was only about 7 square microns. And so you got to get those EM slices just right to be able to catch that. So the slices are usually under 100 nanometers thick. And so it was actually quite rare to get that exact. So this is unpublished work, but we're going to be submitting this for review pretty soon. Now, what are these structures? We still don't really know. My first thought was that it was autophagy. The short answer is, well, probably not, or at least it's not canonical autophagy. So this was some work that's in review right now by a PhD student, Matt Lemke, shown here. David and Marta were also involved in this project. So what we did is we crossed in autophagy mutations affecting macroautophagy or autophagosome formation, the canonical ATG mutants, crossed them into the FC2 background. And long story short, it really didn't do much. FC2 mutants still died under cycling light. And this is the trypan blue stain showing cell death. So FC2 mutants die. The FC2 ATG double mutants still die. And even more importantly, the chloroplasts still degrade. So here's a degrading chloroplast in FC2 ATG7. It's still blebbing out into the central vacuole. So clearly, autophagosomes and macroautophagy isn't required for this. So what is it? We really don't know. Our best guess is that it's something called microautophagy, or at least autophagosome independent microautophagy, which is a real thing, but is very poorly understood, particularly in plants. There's some putative marker genes that have been proposed. Interestingly, they're all upregulated in FC2. So right now we're thinking this could be this very poorly understood process of cellular degradation called microautophagy, which basically involves structures being pushed into the central vacuole, basically just like this. Okay, so let's, going back to signaling and trying to figure out exactly how these chloroplasts could be targeted for degradation. So we have this nice mutant here, I said FC2, where you can grow it 2 different ways. You can grow it under cycling light, where all the chloroplasts get degraded at once because of singlet oxygen, or in constant light, where you get individual chloroplasts being degraded. but you don't get the cell death. So I do think this is actually, this was a signal because singlet oxygen is a very unusual ROS, and that has a very short half-life. So it can't really move very far. So if any singlet oxygen made in a chloroplast is going to probably stay within that chloroplast, meaning that secondary signals would have to be involved with cellular degradation pathways. And there's a lot of literature suggesting that singlet oxygen has the ability to do this in plant cells. So to try to figure out what these signals could be, I did a genetic screen when I was in Joanne's lab, looking for second site mutations that block cell death in cycling conditions. So we call these ferrochelatase 2 suppressors, or FTS mutants. We got a bunch of them, about 26, made mapping populations, and then did bulk deep sequencing of those mapping populations to rough map the causative alleles. This is where all of them fell on the 5 chromosomes. The ones in red we've cloned, and we know what they are. So we're working through these mutants to try to understand the signaling pathway, and they're starting to fall into different classes. So we have classes 1, 2, and 3. Class 1 is, I guess, the boring one. These are mutations we think are just reducing singlet oxygen stress, so they're not really involved in signaling per se. Class 3 are quite interesting. These are ones I'm not going to talk about today, but these are ones that we think are working downstream of chloroplast stress, and they're proteins that are actually outside they encode proteins outside the chloroplast. But class 2 is what I'm going to spend the rest of this morning or this evening talking about. These are proteins that we think are actually involved in generating the stress signal in the first place within the chloroplast. And this is work that we've just published this year, led by Cameron Alamdari, who's a technician in the lab, and also Karen Fisher, microscopist, and David and Stigna have also been involved in these projects. So, we started going through these mutants. I'm showing you pictures of FTS3, 32, and 38. We chose these because they seem kind of similar. They were all very pale under constant light compared to FC2 right here, but they survived under cycling light conditions. Here's a trypan blue stain to show you that FC2 stains blue because it's dead, and these double mutants don't. We mapped them. 3 and 38 are allelic. They were in a gene called PPR30, which is a pentatrichopeptide repeat containing protein. I'll tell you what that is in a second. And FTS32 was a mutation in mTORF9, which is a mitochondrial termination factor. And I'm not showing it here, but we did all the appropriate complementation and allelism tests, so we're pretty sure we got the right genes. Okay, so PPR proteins, if you don't know what those are organelle localized proteins involved in post-transcriptional regulation. So they bind specific transcripts and affect their expression either through promoting stability or degradation or splicing, translation or editing. They can basically do anything involved in regulation. mTORFs are similar. They're called mitochondrial termination factors because that's where they were originally discovered, but they are also in chloroplasts. They do affect transcription termination, but they can also affect gene expression in other ways. And again, by binding specific transcripts or in this case, proteins. And so both of these proteins probably are affecting gene expression in the chloroplast, and we did localize them there. So we know for sure that they are chloroplast proteins. And as you might expect, these mutants have defects in chloroplast gene expression. So we checked that by qPCR, and this is just showing some genes that we checked in the chloroplast. A lot of them, but not all of them, are reduced in the FTS mutants. And importantly, this did not lead to a reduction of singlet oxygen in most of them. So FTS3 and FTS32 both still had high levels of singlet oxygen. So that was saying to us that this really was affecting the signal coming from chloroplasts, and that full chloroplast gene expression was required for that signal. Now, a related mutant, this paper just came out last month. This is FTS39. So again, this one was also pale, but it also suppressed FC2 in that it was able to survive light cycling conditions. You can see that down here where FC2 stained blue, FTS39 didn't. This one, but when we cloned it, we were surprised to find it was affecting a cytoplasmic enzyme involved in nucleotide metabolism. So this is CTPS2, or cytidine triphosphate synthase 2, which encodes the rate-limiting step of de novo CTP synthesis in the cytoplasm. This was a weird mutation. It was affected the splice site, leading to a cryptic splicing, which led to a cryptic splice site, which then produced a transcript missing 6 bases that eliminated 2 essential amino acids. So this mutant also reduced plastid transcription, or chloroplast transcripts, but it also reduced chloroplast DNA levels, which made us think, oh, maybe it's actually affecting d-CTP pools for chloroplasts. That's what we think is going on, because if you feed deoxycytidine, which can be salvaged by plants, you rescue all these phenotypes. So now the CTPS2 mutant dies again under cycling light when you feed it deoxycytidine, DNA levels go back up, and transcript levels go back up. So we think ultimately this is doing the same thing as the other mutations, where it's reducing gene expression in the chloroplast, but by this very sort of indirect way, by manipulating nucleotide levels in the cytoplasm. So this was all pointing to the possibility that if you reduce gene expression in the chloroplast, that blocks these singlet oxygen signals. So we tested that directly by feeding different inhibitors to our seedlings. So rifampicin blocks the RNA polymerase and reduces transcription. And sure enough, if you add this to seedlings, FC2 mutants stop dying. You reduce cell death. Right here in red, you reduce cell death when you feed rifampicin, or you can reduce DNA content with nalidixic acid or Cipro. And again, you add these drugs to FC2 and you reduce cell death. So both of these altogether, this is suggesting that full chloroplast gene expression is required for singlet oxygen signals. So here's our model that we're working on right now, where singlet oxygen can accumulate in chloroplasts under various types of stresses, but particularly in our FC2 mutants. That leads to a singlet oxygen signal that's dependent on gene expression in the chloroplast, and particularly PPR30 and mTORF9. So right now we're hypothesizing that these proteins may target specific transcripts that are encoding factors involved in this signal. We also know that you can manipulate this signaling by changing nucleotide pools in the cytoplasm. And I didn't have time to go into this, but CTPS2 is actually part of a conserved clade of enzymes that we think can work together in this fashion, providing nucleotides to chloroplasts. And then somehow, and I didn't have time to talk about this today, somehow this leads to marking chloroplasts for degradation. At least that's our hypothesis. And one possibility could be that these damaged chloroplasts get ubiquitinated on the envelope proteins by an E3 ligase, possibly by PUB4. This is another mutant we got from this is another protein that we got from our mutant screen. And then eventually these get removed from the cell. And again, it seems that it's autophagy independent, or at least macroautophagy independent. So we're thinking that it's possibly a microautophagy pathway. So right now, this is as I said, this is the model we're working on, and we're focusing right now on trying to understand what these signals are coming from these chloroplasts and what the targets of PPR30 and mTORF9 may be that would allow this signaling to occur. Okay, so a few people to thank. These are the people in my lab that I like to thank everybody here. A lot of the work I showed you was done by Cameron right here and Karen. And I need to also thank the Department of Energy, which has been funding this research while I was in Joanne's lab, and I have my own grant with them right now at the University of Arizona. And with that, I'll thank you for your attention. And I guess it's time for questions.
Okay, thank you, Jesse. That was a fabulous talk. I really enjoyed it.
Thank you.
So since I've got the microphone, I wanted to first ask a question of Monika. Have you, Monika, or has anyone in the field of stress granules thought of using flow cytometry and sorting to identify changes in the stress granules or to directly isolate them?
I think so far people, you can hear me, right?
Yes, yes.
I think so far people focus on more direct methods to pulling them based on the bait. So I know that previously the major attempts were to do the differential centrifugation. So would be also separation based on the mass, but it always leads to different complexes being pulled together with the stress granules.
Mm-hmm.
So I think that's why people move for more direct. So if they are stress granules based on the cytoskeleton, which is formed by the specific proteins, now they are pulling like this.
I was impressed with the fact that the fluorescent protein labeled granules seemed very discrete. They were easy to see and they were point sources of light. So yes. I think it would be, and since it's so easy to homogenize, make a homogenate from plants, just chop up the plant, it should be very easy to take one of your transgenics and run it through, run the supernatant through the flow cytometer to see what it looks like. And Jesse, I would say the same with your system. Have you considered looking at your chloroplasts at various times after induction of changes, reactive oxygen species and so forth, to be able to look at the fluorescent spectra of the chloroplasts and see what happens, and then isolate chloroplasts that have different patterns of changes in their intrinsic fluorescence properties?
Yeah, this was actually something that we were, the postdoc in the lab, Snigdha, was actually trying to tackle before the COVID hit. But looking at the changes in the proteome during after singlet oxygen stress, and so yeah, that was something that we still plan to return to.
How many chloroplasts do you think you would need to collect to get a signal, whether it be at the RNA or the protein level?
Not too bad. I mean, it's a technical thing. I mean, it's just you can start with a few grams of leaf tissue. I think would probably give you enough chloroplasts to do whatever sort of downstream.
Yeah, now I'm thinking in terms of millions of chloroplasts.
Oh, the number of chloroplasts.
Yeah.
I don't know. Yeah, I don't know the number. I mean, the good thing is these things are chock full of RNA and protein. So yeah, maybe you wouldn't need that many, but yeah, I'm not sure how many you would actually need.
Yeah. Okay. All right. I'll open the floor up for questions.
So just to comment, David is a expert in flow cytometry. So
there's no better technique.
Jesse, is there a I mean this you call it autophagy?
Yep.
Is there do you think you could make a screen for that, like a fluorescent screen or something to identify a gene that's expressed at that time, and then put a fluorescent tag on that, and then
that's what we were hoping to find with the proteomics, that these chloroplasts would be tethered physically to structures involved in moving them to the central vacuole, because in these microscopy images you can see the chloroplast envelope is just right up against the tonoplast, the membrane for the central vacuole. And the way these are supposed to work is that there's receptors that are interacting with one another. And so what we were hoping is that we pull down these degrading chloroplasts and we'd look at the proteomes and we'd see tonoplast proteins, or we'd see proteins that shouldn't be in the chloroplast that would maybe help point towards these receptors. And then we would tag those and see where they are. But the challenge is, not much is known in plants for how this works. And it does happen in yeast, but it's not clear how conserved that might be. So the other thing is you could try to find the yeast orthologs and work that way. But yeah, it's just not, there's really little known.
So have you it's a FC2 mutant, is that correct?
Yes. Mm-hmm.
Have you, this is trying to merge yours and Monica's research, but have you guys looked at stress granules in FC2 mutants?
No, we haven't. And I think that would be a really interesting thing to do. I bet they're there. And then in full light, or do cycling the light and see what's going on. That would be, that'd be quite exciting to. Yeah, I think that's a good idea. And Monica, it sounds like you're interested in looking at high light stress, which would probably be even more similar to what we're looking at, because high light tends to make singlet oxygen over hydrogen peroxide. So you would potentially get different sorts of effects. That could be, so there could be, I'd definitely be interested in seeing if we see similar sorts of structures in under our types of stress.
Yeah, that was a good suggestion.
That would be pretty cool, I think. To look at that.
Yeah.
More questions?
Salim has a question.
Yeah, thank you very much, Jesse, for this excellent talk. I like the story so much. I have a stupid question. In the model that you presented on your last slide, you assume that disturbing the transcription would somehow reduce the production of the signal, of the retrograde signal that goes to the plastids. But it's also possible that when you reduce the transcription and replication in the plastids, you just get less active plastids. And this means lower production of singlet oxygen.
Oh, yes. That's a good question. So that's what I originally thought, but we went in and measured the singlet oxygen and it's still high. And not only that, the tetraprol pathway, which is generating the singlet oxygen, also was not affected. And so right now we're thinking this is that the chloroplasts are still stressed. They're still making singlet oxygen, but they're not doing the signaling. And so that's why we have that model suggesting that it's a product of the plastid genome that needs to be present in order for this signaling to occur.
The other question is, did you see any interference, any relation overlap with cyclocitral signaling?
Beta cyclocitral? Okay, yeah. So, right. So that's a good question. So beta cyclocitral is a volatile oxidation product of carotenoids that's made during singlet oxygen stress. We have not specifically looked. However, the Michel Havot's lab that in France that investigates that, their thinking is that it is completely a transcriptional signal, that it does not regulate cellular degradation, that it's a signal to the nucleus it's a topic retrograde signal. So that could still be being made in our mutant, and that could still be affecting gene expression in the nucleus, but we haven't actually looked.
Thank you very much.
Yeah, thank you.
Do we have more questions? Just go ahead.
Jesse, I have a question about the constant light versus cyclic light. Why would be more oxidative stress under cyclic conditions versus constant light conditions?
Okay, so yeah, Zhang is asking what's the difference between cyclic light and constant light for our mutants. So it is a oddity to the FC2 mutant that's not necessarily necessarily true in wild type, but the pathway, the biosynthetic pathway FC2 is in, gets activated at dawn by many different levels. It's a transcriptional activation, it's a post-translational activation. So that pathway gets turned on very quickly. And if FC2 is not there, its substrate quickly builds up, because probably for kinetic reasons, that that molecule can't get turned over fast enough and sort of quickly accumulates. And constant light, everything's steady state. And so you just don't get that abrupt accumulation of the protoporphyrin 9.
Okay,
So yeah, it comes down to the kinetics of the biosynthetic pathway.
So this is just particular to this particular mutant.
It is particular to that mutant. However, under natural light, plants don't really like it if you go from complete dark to light. That's not a very natural thing. And that actually does lead to ROS production in the chloroplasts. Not enough to necessarily kill everything, but that actually is a stressful situation that makes singlet oxygen in the chloroplasts.
Okay, thank you.
Yep.
So could you control weeds by doing that? Flashlight at them?
Control weeds?
Yeah, just have LEDs out in the field and just flash the light down.
Yeah, you kill everything else. You'd harm everything else too.
Well, direct them downwards, I don't know.
But no, but that's, I mean, but that abrupt changes in light intensity is usually, a stressful thing. Mm-hmm. Again, not necessarily leading to massive cellular degradation, but it's something that the natural protective mechanisms of chloroplasts can deal with.
Going back to flow sorting, I think you could sort out those individual damaged chloroplasts at various times and then look to see what's in them in terms of metabolites, transcripts, and proteins. That's why I'm interested in how many you'd have to sort to actually get a signal. So I'm going to have to
maintain their integrity during the sorting process.
Oh, that's not an issue.
They might fall apart, mightn't they? Because they all look very delicate.
Okay.
David,
Yeah, my feeling is that the chloroplasts tend to be like bullets compared to, let's say, a protoplast.
Yeah.
So
absolutely. Absolutely.
That might be worth trying then.
But those,
that'd be easy.
Yeah. Those pictures Jesse had of degrading chloroplasts, they all sort of looked, I don't know, they didn't look very as bullet-like as a.
No,
yeah, that's true. The one with the hernia, they're a little odd.
Could I just ask a question?
Go ahead, please.
It's just pick up on your comment that this business about flashing light on the weeds. It's not completely silly thought, because you could, because especially with machine with machine vision stuff now, if you have them recognizing the weed in the dark, patrolling the field, and then recognizing the weed and then flashing the light on them, it may well be something. I know it sounds a bit sci-fi at the moment, but it's not completely insane as a thought.
Trust the Brits to come up with something insane.
Yeah, well, they're thinking about it for insects, because insects, sensitive, obviously, if flash UV at them, you can explode them, but also blue light, which is, there's a paper in, it wasn't in Nature, but it was in one of the Nature family journals, Nature Comms, just a few months ago, with blue light being lethal to insects. And so they were suggesting we zap the insects with blue light. Anyway, sorry, I should go on to my question, which was that this is for both of you, actually, Monika and Jesse. And I may have missed stuff in your seminar, and I really apologize if so. But I mean, I saw you both describing really interesting processes and beautiful biochemistry, beautiful cell biology. But I wasn't sure I saw you relating the processes that you were describing in response to stress as being relevant to the adaptation to stress. In other words, if you alter manipulate the processes? Do you manipulate the tolerance of the plants to heat stress or whatever the highlight stress, whatever it is that you're interested in?
Okay, maybe I go first. So in general, let's say stress granules, they are not well studied in plants, but there were few reports showing that if there is overexpression of a protein, which is a key component of stress granule, it increased the tolerance to the heat stress. So apparently it helped to recruit the right candidates to go to the stress granules to facilitate the response. But of course, the field is just starting, so that's why we would like to understand more how does it work and based on what is the selection happening. But in general, that is the function of stress granules, that they have these properties. They can recruit different components and therefore like speed up the recovery after the strong stress and also increase the tolerance for the stress.
Great, thank you.
And then I'll say, hasn't been our focus yet, but something that we want to start looking at more. But we have little bits of data here and there. For instance, some of the suppressor mutants we have picked out, if you put them in the wild type background, they're more resistant to excess light stress. So you get less cellular degradation in high light and cold temperatures that make singlet oxygen. And then a really interesting phenotype is the ubiquitin E3 ligase we got out of our screen, PUB4. We've shown that it senesces early, and particularly in carbon and nitrogen limiting environments. And so we think that if you can't turn over your chloroplasts, you're bad at nutrient distribution.
Sure.
And what's cool is that this is with a collaboration. We showed that this is particularly bad in autophagy mutants. So we think that there could be a parallel pathway to autophagy in turning over chloroplasts during starvation.
Sure.
But still, it's all kind of, as Monika said, this is all kind of new stuff, and we're trying to figure out exactly. Yeah.
No, I think it's great the way you guys are both sort of bringing together cell biology and biochemistry. I think it is a new frontier and very, very interesting. If you can then relate it to whole plant function or adaptation, then wow, that'll close the whole thing. So, at least for me, yes. Thanks a lot.
Of course. Thanks.
Ravi, I see you had your hand up.
Yes. Hi. I just had a quick question for Monika regarding the stress granules. What is known about stress granule accumulation when plants are acclimatized and then they're exposed to stress? So this is it in this case, it looks like there's a sudden exposure of stress and then that's how plants respond. But it is known that if you acclimatize plants, then they can deal with eventual stress better. And in that context, what is known about stress granules? And I have another question on stress granules. I'll wait to ask you.
Okay, so I think there is no study published yet what is happening when you expose plants to the, let's say, mild stress, you acclimate them and then you apply the strong stress. But I know that few groups are working on that. And what they observe that the stress granules in the major stress, they are formed faster. So it seems that the plant can respond faster to stress. But yeah, as I said, that is not published and still needs to be investigated.
And the other, sorry, go ahead.
No, that I would also expect that it would rather facilitate the response. So one of our projects also will be to do this kind of pre-treatment and then the major treatment of stress and see also if the composition will be different of stress granules which appear in the mild stress and in the, let's say, the big stress. Because I think there will be also differences in that.
Okay. And the other question, I have a little different take on it, is that I'm interested in the evolution of these stress granules. So if you were to look at the normal plant development, because I always think that it's probably being used somewhere else and then it's being recruited in terms of stress. Is it anything known about such granules formed in any aspect of plant development? Because even during normal course of plant development, there's a lot of dynamic changes in the environment to the plant. Perhaps they are being used there, and then under terms of stress, they are efficiently recruited to deal with it.
To be honest, I don't know. I would expect, so let's say the formation of stress granules also rely on like pre-complexes. So there are many complexes that are formed even in normal condition between few components. So might be that without the particular stress, there is already pre-formation of the complexes, which might be used for the stress granule formation. But I'm not aware from the evolutionary point of view. No, I really don't know.
No problem. Thank you. And Jesse, quickly along those lines, are there any developmental phenotypes at all for FC2 mutant, for example?
Yeah, that's a good question, Ravi. Under permissive conditions, not really. It's just a bit delayed to green. That could very well be just because it's a heme auxotroph as well. And so the cytochromes in the chloroplast are maybe it takes longer for chloroplast biogenesis to occur. But other than that, no, there's nothing particularly different about them if you baby them under permissive conditions.
Okay. And also you guys have tested the light to induce the stress, but are there other stresses you have checked where, because I would imagine the chloroplast will be also responding to other stress like drought or heat.
Oh yeah. We've looked at cold, and the reason for that is that naturally photosynthesis will produce singlet oxygen in the cold because you're slowing down the carbon reactions. And so you don't have enough NADP as an electron acceptor during the light reactions, and you build up energy in the photosystems and you make singlet oxygen. So that's just an alternative way to make singlet oxygen is high light at 4 degrees. But we've purposely stayed away from heat because heat makes peroxides, and we're trying to keep it as simple as possible by making only one type of reactive oxygen species. But under natural stress, you're usually making lots of different things. And I doubt chloroplasts normally just see one type of ROS, but so we're trying to simplify it, but so that's why we do it the way we do.
Great, thank you both.
Thank you.
Okay, do we have any more questions? We have a couple more minutes.
I just had one question to Monika. Is there a stress granule system in monocots that people are working on, or is it all in Arabidopsis now?
I think for now it's just Arabidopsis.
Oh dear.
That's
So
enough.
our idea is to move a bit to rice, but yeah, so far the only research, there are just few publications in plants, and everything is based on Arabidopsis.
Okay.
So everything's open then. That's great.
Yeah, exactly.
Yes.
Yeah. Actually, I like Jesse's comment just then about peroxide and heat and superoxide under high light load temperature. I actually didn't know that. That opens up some pretty interesting possibilities, actually. I mean, look, you guys have to obviously let all this happen organically. It just sets my brain off with interesting manipulations that could be relevant and complementary to your current research, Jesse. That's all. Because heat's a pretty big issue here.
And here.
Yeah, fair comment, actually. Yeah.
Okay, are there any more questions? All right, well, we've actually ended on time, which is a wonderful situation. Thank you, Jesse and Monika, for staying within the remit. And it was really a pleasure to listen to your talks. They're very well produced and very exciting work. So thank you, everybody. Let's give some round of applause to our speakers. And
Thank you.
And I'll look forward to talking with you next week in this seminar series, which I think is really a wonderful thing. And thanks, Rod, for organizing it.
Well, thank you. Thank you, Jesse and Monika. It's been great, and we'll see you next Tuesday.
Okay, thank you.
Thank you very much, everyone.
Okay, thank you.
See everybody.